We are going to learn in this session about the process of the formation of gametes. Now, gametes are extremely important cells in organisms that reproduce sexually, because these are the cells that not only carry the information forward from one parent, but also when they fuse together, they give rise to a new organism, a product of sexual reproduction. Now, today we will start off with the process of gametogenesis, and gametogenesis simply means the birth of gametes. Genesis is birth, and gamete is a sex cell. Now, we'll start off with gametogenesis of sperms, and the process of formation of sperm or the birth of sperm is also called spermatogenesis. Now, before we get into spermatogenesis, let us rapidly recall where does this process takes place. Now, this process takes place in the testis in males, and here we have the diagram of the testis. Now, there are a variety of substructures present inside the testis, but what we are interested in is where inside the testis are the sperms made, where spermatogenesis takes place. Now, spermatogenesis takes place inside these highly coiled tube-like structures found inside the testis, which are called the seminiferous tubules. And it is astounding to know that the length of the seminiferous tubule in each testicle ranges from 90 meters to 120 meters. 90 to 120 meters. Such is the length of the seminiferous tubule in each testicle. Now, for all the good reasons, the longer the seminiferous tubule, the greater is the area for the formation of the male gametes, and male gametes are produced in vast numbers. Now, if we take a section of the seminiferous tubule, when I say a section, what do I mean? Is, let us say this is the seminiferous tubule. If I cut and I take this slice out from here, if I take this slice and look at it exactly from above, the bird's eye view, I get a cross section. And if you cut a seminiferous tubule here, this is how the cross section looks. Now, inside the cross section, you can see the hollow center, and this hollow center is called the lumen or the hollow central space. And around the lumen, we've got wide variety of cells, and eventually at the outermost region we have the border or the wall of the seminiferous tubule. Now, we are going to just take a little bit, a part of this whole section of the seminiferous tubule to understand it much better. So what we are going to do, we are just going to understand, we are only going to take this much of the part of the whole of the section of the seminiferous tubule, and this is how it appears magnified. Let us magnify it just a little bit more, and there we go. So we've magnified it like really well. Just that's it. And now let us drag it down. So the wall of the seminiferous tubule is made up of a single layer of cells, and this is an epithelial layer. Now, just beneath this wall we have got specialized germ cells. When I say germ cells, I do not mean the disease causing germs. Germ cells and somatic cells are the two types of cells that are found in the body of all types of living multicellular organisms. Germ cells have only one function: they simply divide. So in case of plants, the meristematic cells are the germ cells. In case of animals, we have got these germ cells or stem cells. So we got germ cells here, and their sole purpose is to simply divide mitotically, which means when they divide, they give rise to diploid cells. So there are a large number of germinal cells. Now, what happens to these germinal cells, which are present exactly close to the wall of the seminiferous tubule? They undergo mitosis, and they produce large number of cells which are just like them. Now, these germ cells are also called spermatogonia. They are also known as spermatogonia. So these germinal cells are also known as spermatogonia. When spermatogonia, they undergo mitotic division. You see, mitotic division will produce, these are diploid cells. Undergoing mitotic division, each cell will produce two diploid cells. So germinal cells or spermatogonia produce more spermatogonia. Now, a large number of these spermatogonia will simply add up to the layer of germinal cells, because the older germinal cells eventually will die and they have to be replaced, and they are replaced by these germinal epithelial cells. But a few of these spermatogonia or the germ cells will will make something known as a primary spermatocyte. Now, what is the difference in a primary spermatocyte and a germinal cell? Is that primary spermatocytes further divide. Well, the germinal cells do divide by mitosis, but primary spermatocytes undergo a different kind of a cell division, which is called meiosis. So far, germinal cells have the ability to divide and add up more germinal cells by the process of mitosis, whereas some of the germinal cells, which are called primary spermatocytes, have the ability to undergo meiosis. Now, these primary spermatocytes which are undergoing meiosis, what happens with them? Let us see now. Now, the primary spermatocyte, these are diploid cells, as has been mentioned here, which means they have a pair of homologous chromosomes, one from the father and the other from the mother, and which is shown here in this particular image. We've got two pairs of chromosomes from the mother and two pairs. Basically, these are all doubled up. Okay, just before meiosis, I hope you remember what happens prior to meiosis. The amount of DNA doubles up. So the cell is already diploid and it has half the genetic content from the mother, half from the father, which is 2C, and just before a cell has to undergo meiosis or mitosis, this content of DNA doubles up, and this condition is called the 4C. So the cell remains diploid even after the DNA is doubled up, but the amount of DNA has been doubled up, which is represented by 4C. So the primary spermatocyte, which is diploid but has double the content of DNA, now undergoes the first meiotic phase. And what is the end result of meiosis 1? We get two haploid cells, but the content of DNA in both of them is 2C, is halved. You see, here the content of DNA was 4C and here it is 2C. It has been halved, but these two cells are haploid. Now these two haploid primary, this primary spermatocyte, when it underwent meiosis 1, the two cells that are formed are called secondary spermatocytes, and they are haploid. So these two are haploid. But meiosis does not end here. What is the purpose of meiosis 1? Meiosis 1 achieves the purpose of recombination, or crossing over takes place here in meiosis 1, right, during the prophase 1, and genes are exchanged between the chromosomes, between the homologous chromosome, the father and the mother's chromosomes. The genes are exchanged and we get new variations here. And for that particular reason, each of them is considered a haploid cell. Anyways, there is more to it, but we won't touch that topic here. Now, after this particular stage, each secondary spermatocyte undergoes meiosis 2, which is greatly equivalent to mitosis. And what we get is the ploidy remains the same. You see, mitosis, one of the hallmark properties of mitosis is that the ploidy never changes. So haploid cell give rise to haploid cell. Each cell divides into two. Haploid cells are formed, 4 haploid cells, but now the amount of DNA is halved, so each of them has a single set of chromosomes, right, not a pair. And so we get 4 haploid chromosomes, 4 haploid cells, and these 4 haploid cells are called the spermatids. They are called spermatids. So secondary spermatocyte undergoes meiosis 2 and forms 4 haploid spermatids. Now these spermatids are immature, and what happens to these spermatids is that there is this particular large cell, these cells which are present in the seminiferous tubules. These cells, you see here, this cell right here in this space around, so this particular cell is called the Sertoli cell, and this function of the Sertoli cell is to nourish and lead to the maturation of these spermatids. Now you see, spermatids don't have the tails, and the tails have to be formed, then they have to be made into functional sperms or the spermatozoans. Now these spermatids attach themselves to the Sertoli cells, and the Sertoli cells then bring out changes. They nourish them, they also bring out changes, they transform them, and the spermatids are differentiated into cells called the spermatozoa, or in simpler language, the sperms. And here we have them, right? And we have them here, right? I hope you can see that. So this is the process of spermatogenesis, or the process of formation of sperms. Now these spermatozoans eventually are released into this hollow vacant lumen of the seminiferous tubule, and when sufficient amount of sperms are made, the process of spermatogenesis comes to a halt. It stops, or at least the speed of spermatogenesis is reduced. Now, how does the body know when to make sperms, when not to make sperms? Is something that we'll learn now in our next topic, which is here. Feedback regulation of spermatogenesis. What controls, how is spermatogenesis controlled? Now, I hope you remember that human beings do not produce sperms. The human males do not produce sperms the moment they are born. They produce sperms once they've hit puberty, right? So these are certain changes, hormonal changes that bring about, that lead to our maturation, sexual maturation. Now, there are certain hormones involved in sexual maturation. And what are those hormones? The important hormones that are involved in spermatogenesis are GnRH. This is also called gonadotrophin releasing hormone. And then we got another hormone called LH, or luteinizing hormone. And the third is FSH, which is called follicle stimulating hormone. And the last hormone is called testosterone. Now, there is another term which is known as the androgens. Now, androgens are all those hormones which are not exclusively but are largely produced by the males, right? So testosterone is produced in larger amounts by in the bodies of the human males, and that's why testosterone is an example of an androgen or a male hormone. So things to remember: the process of spermatogenesis is under the control of these four hormones: the GnRH, or gonadotrophin releasing hormone; LH, luteinizing hormone; FSH is follicular stimulating hormone; and the last one is the testosterone. Now, how does this process start? Now, before the human males have achieved their puberty, before we have before we have become sexually mature, before puberty, inside of our body, the level of these hormones called the luteinizing hormone and follicle stimulating hormone is very low. It's not very high, and these hormones are LH and FSH are required by the body for the process of spermatogenesis. But the moment puberty is hit, certain hormonal changes take place in the body, and one of those important changes is the greater amount of release of GnRH, or gonadotrophin releasing hormone. Now, where is this hormone produced? Now, hypothalamus, I want you to think for a moment about the structure of the brain. Inside of the human brain, there's this structure called the hypothalamus, and in this structure called hypothalamus, this particular hormone is produced. So at the time of puberty, the body starts to produce larger amounts of GnRH. And GnRH, and then there is another part of the body which is called the pituitary gland. Now, this pituitary gland is a neuroendocrine gland, which means it is partially a structure of the nervous system. It's a part of the nervous system partially, and the remaining half of this pituitary gland is an endocrine gland. Endocrine, not endocrine. It's an endocrine. So let's not anyways write it. I hope this is clear to you. So pituitary has a dual role. It is a part of the nervous system as well as a part of the endocrine system. So this particular hormone, which is released by the hypothalamus, GnRH, then goes to the pituitary. It is received by the pituitary. This particular hormone is received by the pituitary, and as a response to this hormone, the anterior pituitary then releases two hormones in larger amounts, the luteinizing hormone and the follicular stimulating hormone. These two hormones are now released by the anterior pituitary. Let's use a color. Let's use green for luteinizing hormone, and let's use yellow for the follicular stimulating hormone. So these two hormones are now released in larger amounts. It is not that these hormones were not released prior to puberty, but now they are released in larger amounts. Now, what do these two hormones do? These two hormones are released into the blood. And the blood, and that's why this is an endocrine gland. Endocrine glands release their secretions into the blood. Now, the luteinizing hormone and the follicular stimulating hormone. Let us look at luteinizing hormone. The luteinizing hormone moves throughout the body. Now, remember this particular, all the hormones are going to circulate throughout the body. They are going to come in contact with all the cells, but not all the cells will respond to them. Cells that have receptors for them will only respond, and the cells called the Leydig cells specifically and will respond to luteinizing hormone. Now, first of all, let us see where Leydig cells are, and let us magnify this image. Now, this is a section of the testis, and here I've hugely magnified it. Let us magnify it a little bit less here and here. Now let us look at the Leydig cells. Now Leydig cells, here we have the Leydig cells. Now this is a section of the seminiferous section of the testicular lobule, and inside this lobule we got large number of seminiferous tubules. So this is one seminiferous tubule, this is another one, we can see a bit of the other, Now these are the seminiferous tubule, and all this space around it is called the interstitial space, and this space has blood vessels, as you've studied in your previous classes. And there are also these cells here. You see this cluster of cells? These cells are called interstitial cells or Leydig cells. Now, these are the Leydig cells. And what happens when the luteinizing hormone is received? See, all the cells are going to come in contact with these hormones. But Leydig cells will react to them, will undergo certain metabolic changes as a result of receiving these hormones, this particular hormone. And what does the Leydig cells do? Leydig cells start producing androgens. And what are androgens? Androgens are the male hormones, male sexual hormones. And one example of androgen is testosterone. So Leydig cells will start producing androgens, in particular testosterone. So this is, if you write testosterone or you write androgens, it is perfect, but it's better to write testosterone and androgens both. So Leydig cells will start producing testosterone and androgen. What does this testosterone do? The testosterone, this particular hormone, has the ability, now it will seep from in here and it will go, let us magnify just a bit, here and here, let us see. So this testosterone hormone will now move into the seminiferous tubules, and here what they will do is that these particular cells, the germinal epithelium or the spermatogonia, let us go back to the previous page and see. Now the testosterone will instigate these cells, the germinal cells or the spermatogonia, to start multiplying. They will start multiplying, they will make primary spermatocytes. The testosterone hormone will also instigate the spermatocytes to undergo meiosis, and as a result of the entire process we will get spermatids. And so this is the function of testosterone. So testosterone basically initiates cell division, both mitosis and meiosis inside the seminiferous tubule. Now let's come back and have a look at the next step. Now the other hormone called the follicular stimulating hormone or follicle stimulating hormone, what it does, this hormone acts on the Sertoli cells. And where are Sertoli cells? Let's get back and look inside. Have a look at this cell. This is the Sertoli cell. Follicle stimulating hormone, what is the target of this hormone? The Sertoli cells. And what does it do to the Sertoli cells? This particular hormone FSH tells the Sertoli cells to facilitate spermatogenesis. Facilitate is to help. And how do Sertoli cells help? They start to nourish the spermatids that are formed. They will start to care for them. They'll act like mothers and they will feed them, and they will release certain enzymes and certain chemicals which will transform these spermatids to generate flagellar tails. They'll mature their internal structures and they'll make them ready for their future journeys and functioning. So follicular stimulating hormone does this. It stimulates the Sertoli cells to take care of the spermatids and transform them into spermatozoans. Now I hope these two functions, the functions of the luteinizing hormone and the follicular stimulating hormone, is clear to you. Now as long as now a lot of these hormones are being produced and the number of gamete is now increasing, so the amount of number of gametes or the male gametes, the sperm, starts to increase within the seminiferous tubules. Now if this process keeps on happening and happening and happening, a huge number of spermatozoans or sperms will be formed. Now if they are not used, then this becomes a wasteful process. So the body has not only one function, it does not have to spend its entire energy only towards the formation of sperms. So once a certain amount or a certain number of sperms are formed, this process must be stopped. Isn't that rational? To conserve energy, once we've made a certain amount of a certain thing, we do not make excess of it, because that is a wastage of time, energy, resources. And so the body has evolved its own ways of dealing with excess or waste or wasting of a certain process. And this is achieved by a process called the feedback mechanism. Feedback mechanism. This is how the body uh, brings about changes or regulates processes by feedback mechanisms. It's just like this. When you give me a feedback, I'll change my behavior. For example, if you tell, if you ask me to teach you in Hindi, I'll start doing that, right? So that is called a feedback mechanism. Now, how does feedback work here? Now, Leydig cells release testosterone once luteinizing hormone is received by Leydig cells. When the amount of testosterone builds above a certain limit in the body, so testosterone will be present in the blood at all the times. But when the amount of testosterone crosses a certain limit, I do not know the exact number, but let us assume, let us say it's 100 nanoliters of testosterone in, let us say, 1 liter of blood. If 100 nanoliters of testosterone, 1 liter of blood, if the amount is exceeded beyond this, what happens that the testosterone now exerts a negative feedback on the hypothalamus and anterior pituitary. What it means is that testosterone tells the anterior pituitary, see, my amount has exceeded beyond my fixed level, and now what anterior pituitary must do, you should stop producing LH and FSH. Not only this, the testosterone also tells the hypothalamus to stop making or reduce the amount of GnRH produced. So anterior pituitary and hypothalamus, on sensing excess testosterone in the body fluids, reduce the amount of GnRH and LH and FSH. So obviously, if the amount of GnRH, gonadotrophin releasing hormone, if its quantity reduces, then certainly it will not stimulate anterior pituitary to make a lot of LH and FSH. And anyways, testosterone is also telling the anterior pituitary, it is telling the hypothalamus, don't make this. If this is not made, GnRH is not made, there will be lesser LH and FSH. But testosterone is also telling anterior pituitary to reduce the production of LH and FSH. Now, if LH and FSH are produced in lesser amounts, Leydig cells will not produce testosterone or produce less testosterone. And if there is less testosterone, there is less spermatogenesis. And if there is less FSH, there is lesser caretaking by the Sertoli cells of the spermatids. And basically lesser maturation of the spermatids to spermatozoans or sperms, right? So this is the feedback mechanism. And also the sperms, the higher concentration, when a large amount of sperms are formed, this process also, the sperms also end up releasing a hormone called inhibin, and this hormone inhibin acts on anterior pituitary and also tells the pituitary to do the same. Anterior pituitary, not the posterior, but the anterior pituitary, to produce less and less of LH and FSH. And so you see, if there is a rise in the amount or the number of sperms beyond a certain level, if there is a rise in the amount of concentration of testosterone beyond a certain level, both the testosterone and the products of spermatogenesis, which is inhibin and testosterone, both of them tell the hypothalamus and anterior pituitary to reduce their secretions, thereby reducing the process of spermatogenesis. I hope the process of spermatogenesis in the testis, how is this formed? What are the different stages? What particular cell undergoes mitosis? What particular undergoes meiosis? And what is the function of Sertoli cells? And blah blah blah. And also, how is the feedback regulation of spermatogenesis executed by the body is clear to you. Thank you.